Today we're going to be talking about the experiments of Nirenberg and Matthaei and a later experiment by Nirenberg and Leader. The purpose of these experiments is they want to determine which RNA sequence coded for which specific amino acid. In the Nirenberg and Matthaei experiment, they used a DNA-less cell-free suspension, which is just a solution containing all of the normal organelles of a cell without the DNA or the cell body. They used the homopolymer poly-U as a control RNA template, and they also used 20 amino acids. 1 was radioactive and 19 were normal. The way that they did this is they added the homopolymer poly-U into the DNA-less SAP, along with all 20 amino acids, and they were all attached to their appropriate tRNA. 1 amino acid was radioactively labeled with carbon-14, whereas the rest were normal. The mixture was then incubated, and then the protein was extracted and radioactivity was measured. They repeated this study 20 times, each time radioactively labeling a different amino acid. If the extracted protein was radioactive, then the labeled amino acid must have been what was making the protein. In other words, the codon must code for that amino acid. In their first experiment, phenylalanine was the radioactively labeled amino acid. In other words, the poly-U chain produced a protein chain of the amino acid phenylalanine. They also used chains of poly-C, poly-A, and poly-G. Poly-C produced proline, whereas poly-A produced lysine, and poly-G just folded in on itself, so the results were inconclusive and it did not produce a protein. The reason they used homopolymers is because mRNA had yet to be isolated at this time. As you can see, this is the steps they took to get to their conclusion. They added the homopolymer poly-U to the cell-free suspension. Then, they incubated it at 37 degrees Celsius. Translation occurs and a protein chain precipitate forms. They filtered the protein and checked it for radioactivity. They found that the poly-U mRNA chain translated to form the protein chain made up of phenylalanine. In other words, UUU codes for phenylalanine. In the later experiment by Nirenberg and Leader, they used ribosomes, 20 charged tRNAs with 1 radioactive amino acid at a time, a cell-free suspension, and trinucleotides. Combining 4 nucleotides into 3-letter codes allows for 64 possible combinations. The way that they did this is they radioactively labeled 1 amino acid at a time in a mixture of 20 amino acids and put the solution through a filter. The ribosome held a known RNA sequence, so the filter let the unbound tRNAs pass through, but caught the tRNA that matched the RNA sequence. The sample filter was then tested for radioactivity. If radioactivity was present on the filter paper, then the tRNA matched the trinucleotide used. In other words, the sequence of bases in the codon was the code for the amino acid carried by the tRNA. They were able to identify 61 different codons. As you can see here, this is an example of how they did it. This is the solution of the amino acid charged tRNAs. They pass through, here's a ribosome containing the known sequence GUU. Valine was radioactively labeled and stuck with the ribosome as the rest got to pass through. They were unbound to the ribosome. They were able to tell that valine was coded for GUU. Thanks for watching!